And as far as tonight, we've got Mr. Stephen Williams, and he's coming from oh so far away here in SoCal, and he received his horticulture training at Mt Sac, at Mount San Antonio College. That's in Walnut. And he's also worked at Descanso Gardens and Huntington Library and Botanic Gardens as garden staff and plant propagator and nursery manager, which was for 8 years, where he's honed in his horticulture skills. And he is affiliated with the UC Master Gardeners program since the 90s. And that's how we connected through Laura Hawley, or was that you, Martha? I think it was Laura that passed this on.
It was Laura.
And thank you, Laura, for connecting us. So Stephen is a professor at Mount Sac. He's in his 15th year. You can update that if that's accurate, Stephen. Teaching 8 weekly year-round classes to adults in home gardening. And really fun having this emphasis on sustainability. He was able to join our meeting last month and has similar content locally taught in terms of organic matter and managing it through compost. So he's got a specific focus on sustainability, organic, edible gardening. And he's even got his own home full of ornamentals, fruit trees, and veggies in raised beds using all these organic techniques. And tonight we're going to be focused on propagation, and we've got some nice visuals that Stephen's going to bring to the table. And thank you so much for joining us, Stephen. Looking forward to it.
My pleasure, guys. I'm glad to be here. Let me change this view here. There we go. And we'll actually start up a PowerPoint here momentarily. And as Ty was saying, I've been teaching at Mount San Antonio College now for 17 years. And as of last Friday, I've actually retired. So my time came here. We actually took 6 months to find a replacement teacher who's now filled in teaching the classes. And I've given everything to him. Although I do plan on coming back next fall for our garden site that we have there on campus. I have a 15-bed raised bed site and a fruit tree orchard that I'm going to come back and be teaching the class there. We're still not open yet. We won't be open to students until the fall. So yes, I've been a master gardener now. I was in the original class here in Orange County, and I will be spending more time with them. This presentation was actually developed for the Los Angeles Master Gardeners, who I've been teaching plant propagation to for, I don't know, 9 or 10 years in person, for 2 years with a recorded session. And then this year I did the Orange County group as well with the plant propagation. So without any further ado, let me go ahead and get things going here. Almost didn't have a meeting today. My internet, Spectrum internet today was out from 8 this morning till 5 o'clock. And I didn't know whether it was going to start back or not. Luckily, it made it. So here we go. Now, you do have, I don't know, Ty, have you sent them the handout that I sent to you? If not, I will post that for you.
Correct. So I did not send that out yet.
Okay.
So
I can actually do that right now. Let me see here if I can.
Will that just go direct into the chat window, or is it a file share? Never actually done
Yeah,
that.
that's what I'm going to do here in the chat area. Let me get this file up here. Oh.
See the option now. I can go ahead and do that as well. Because
why don't you do it? You know why? Because my computer has just changed what it's listing, and it doesn't list my source there that I want. So if you could
pull it up right now and post that in just a second.
And then they could have that to follow along, and something that you can use then in the future as well. So I'll go ahead then and start this up. Now I also have, and as time permits here, I do have a couple of presentations that are recorded of air layering and grafting. So depending on how things go with the time, we'll be able to take those. And if not, then we could always give you those links for later as well. And then I do have one link at the end on tissue culture that we are going to send to you as well. So I said this was first done for the Los Angeles Master Gardeners, and it's evolved over the years, and it's much better than it was. The first recording that I did a year ago, I was pretty green with doing things here, and recording especially. So it was difficult to do a quality production. Today, we're going to be looking at sexual plant propagation with seeds, asexual plant propagation with cuttings, division, grafting, layering, specialized structures, and tissue culture. So with seeds, first of all, let's talk a little bit about seed starting. And I have some resources as well for you that if we don't get to you in the meeting, I'll be sure to get to you later. First of all, the seed itself has a seed coat on it. And in many cases, it's as simple as planting that seed in soil and adding water, and it grows, right? It germinates and grows. We, of course, need to be careful about choosing the right timing that we plant that seed, whether it's a warm season or a cool season plant. Also, we want to be sure to plant it at the proper depth and spacing, and ultimately thin them out to the proper spacing. The days to germinate will usually be on that package of seeds. Also, then you'll notice on your seed package, you'll have information about when the seed package was packaged. You want your seeds to be fresh so that the genetic material is viable and you're going to have strong seeds. As I said, most of your seeds in a seed package are very easy to simply plant, water, and germinate. They grow. Longevity of seeds. How long do they last? Well, here's just a list of a few veggies that shows you typically 3 to 5 years, and some less. And after that, again, that genetic material starts to deplete, and the quality that you do produce isn't going to be as great. The quality of seedlings and the production of your veggies and edibles. So keep that in mind. Storage of your seeds. We can properly store them in different ways. It's oftentimes in a cool, dry place, or it may be under refrigeration. In some cases, it's freezing. In some cases, they will actually take seeds and place them in foil envelopes with nitrogen gas, which extends their longevity as well. So seeds then that don't germinate very easily, that have to have some special treatment, and because they have to overcome dormancy. And this dormancy may be by different factors, environmental conditions that would normally wouldn't be a factor for seeds in a package, but special seeds will have special factors that might be a complex combination of different things, including getting water into that seed germ itself through the seed coat. The fact that some seeds want light, would want to be planted on the surface of the soil. Temperature, some want to have warmer temperatures, some cooler, and we're going to take a look at all these in a minute. Getting gases, oxygen and nitrogen and so on into the seed itself so that it can go ahead and germinate. And then, as I said, that seed coat has to be broken down in many cases because it simply won't allow the water to get in. So mechanically, that has to be broken down. Also, there are hormonal influences that can take place on those seeds that take place at different times, temperatures, and moisture conditions that influence these hormones on those seeds. So, stratification, or scarification, I'm sorry, is where we need to break down that seed coat, and it can be in many different ways. In nature, that seed might fall into the soil in the fall as it leaves that fully formed and ripened flower seed head. It falls to the soil. It then overwinters, and that overwintering period of time, microbial activity might break down the seed coat right in the soil itself, and then come spring, the seed can germinate. Also, it may be that a particular fruit or vegetable is eaten by an animal and taken through their digestive system. The acids in their stomach break down the seed coat. When it's eliminated, fertilizer and all, it will germinate. We can try to duplicate this ourselves by treating certain seeds with either a strong vinegar, which there are more concentrated vinegars, or commercially, they'll actually use sulfuric acid. Now, this is something very dangerous to handle, and for the typical gardener, this is not the right thing to do, but vinegar works well. An example of that would be a toyon seeds, that seed coat that is impervious. So soaking the seeds in vinegar or sulfuric acid for either for 10 minutes or up to several hours, depending on the species. The seeds are then washed, planted, and watered in, and at that point in time, that seed coat will imbibe and take in that water and germinate. So mechanical breaking down of that seed coat, mechanical scarification. It may be filing a seed. It might be sandpaper. It might be nicking it with a knife. Clipping it with fingernail clippers works well. Hammering it with a hammer, like in the case of a nut. Or in some cases, such as bird of paradise, needs a special treatment to break down the seed coat, and that would be a hot water treatment. First of all, that seed, if you've ever seen a bird of paradise seed, it has a little orange hair on it, or orange feather, which is actually an inhibitor, a growth inhibitor. So we want to remove that, and it's also an eye irritant, so be careful not to touch your eyes with mucous membranes. But remove that orange feather, we get a pot of water boiling on the stove, we throw those seeds in, turn off the heat, and let them cool down overnight. The next morning, we plant those seeds, and lo and behold, that seed coat has been broken down. So there's many different ways and special treatments that have been developed and for different seeds. And I'm going to give you some information here in a bit on seeds and special treatments and temperatures for them. So stay tuned for that.
Steve, I was able to upload the handout, so everyone should have that in the chat window. If you want to download it or click on it, it's a PDF file that'll bring up in a browser.
Great. Thank you, Ty. So you have your seeds now that have been scarified, ready to plant, but now we have other treatments that might be necessary. Stratification. And this is where seeds are dormant, and they need to have some sort of, oftentimes, a temperature condition for a period of time to influence germination. So shallow internal dormancy goes away when seeds are dried out, and then they're dried to a certain amount, and then they'll be able to germinate. Cold stratification is where the seeds need to be mixed with either sand or peat moss in a closed container or plastic bag, placed in a refrigerator at about 40 degrees. And usually this is for about 8 weeks that they're kept moist, not wet. And so this cool, moist condition provides this stratification process that breaks down this internal dormancy on these seeds so that then they are taken out and they can be sown and germinate at that time. Warm stratification, on the other hand, would be more tropical or palm seeds that need to be maintained from 68 to 86 degrees. And this may be a long process that may be for up to a year. If these seeds need to be on a heated bench, for example, or bottom heat, at which time then these seeds will break that dormancy and they will grow. Now, containers to do all this in. I personally have best luck with plastic containers because I can control the moisture. When I try using peat pots or clay pots, it's difficult to control that, particularly if you're using bottom heat. They tend to dry out much quicker. Now there are these 72 cell tray, small cells that we can use, and these plug trays. And once they have germinated and grow to a certain size, then we can pop them out and transplant them. And again, I prefer using plastic. And again, that's a personal preference. Commercially, they will oftentimes use a flat. And with the media in there, plant a whole row of seeds. Once they're to size, they will dump these guys out and transplant those individual seedlings. As they're doing thousands, not just a few like you might be. So choice of containers is a personal preference, and you'll find with experience what works best for you. A seed starting mix. Now you can buy commercial mixes, and they're great and a lot easier to use than making your own. But If you were to make your own, a recipe would be something like a peat moss or cocoa peat. Now, I want to differentiate between those and note that peat mosses come from peat bogs that are environmentally something that really should be left in place. We shouldn't be extracting that. We're releasing carbon into the environment, but many of our products do have peat in them. Alternatively, we can use cocoa peat or core peat. And I try to use that whenever I can as an alternative, mixed with either vermiculite or perlite. If it's the peat moss, then we want to use the dolomitic lime to sweeten it up there a bit. With the superphosphate can be added. What this will do is once these seeds germinate, it will give them a strong root system, get that root system going. So we can mix this business up and use this in our containers or in our seed trays, however we're going to use it. And commercial mixes work great though. Now we're going to talk about indoor germination, and then we'll talk about outdoor germination at the end. I like starting my seeds indoors because, again, I have better control. I don't have pests, diseases, climatic conditions. I can control all those indoors and prevent a lot of problems. You always want to use a sterilized container if you're reusing containers or new containers. Your seed starting media should be pre-moistened. You don't want to sow your seeds and then water and have your seeds fly all over the place. So pre-moistening the media will prevent that from happening. Typically 2 to 3 seeds are planted and at the proper depth. And of course, that's on your seed package or on the information that I'm going to give you that you'll have resources to go to. The depth of those seeds is typically 2 to 4 times the seed diameter. And that's a general rule of thumb, but you can get more specific information as I told you from the sources I'll give you. Then we want to cover those seeds. Now we could use our seed starting mix to cover them, or here's a tip for you. I've discovered a product that's actually, instead of using the peat moss, it uses a sphagnum moss as a seed cover. And you'll find out why in just a few minutes here, and it's going to help prevent some problems later. Worm castings could also be used, fine worm castings, and this will provide nutrients to those seedlings as they first do germinate. So labeling, I always want to label. If you're doing individual rows there, you can put little plastic labels right on the container and indicating what is in each of those rows. You'll be happy that you did in the end. It's really important, particularly if you're growing seeds or plants for Master Gardener giveaway or something. You don't want things to be mislabeled. And as well for you when you do your green scenes and the like that you've been doing for so many years, and will come around again next year, we hope. So we've labeled them. We want to provide the proper temperature, whether they are warm season growing seed or a cooler season seed. You have choices of how you're going to keep these moistened and watered. Some folks like to provide moisture from underneath that by osmosis works up through the seed mix. I personally find that a little messy when I'm growing things indoors and doesn't work as well for me as using a spray bottle to mist these with regularly. I discovered something recently and looking for a way so that I didn't have to keep pushing the trigger on a plastic spray bottle. I found a spray bottle here that actually is rechargeable and electric and holds about 4 cups of water. And I simply push the button and it sprays, and I push it and it stops. So when you're doing several flats of seedlings, you're happy to have something like that. And it works really well. So I found that on Amazon, if you're interested. So we want to water these seeds in their respective containers frequently to keep them moist, but we don't want them to be waterlogged. Those seeds could rot if they're too wet. We don't want the surface to dry out. It needs to maintain moist, and that's where the spray works really well to keep that surface moist. If you try watering from beneath, sometimes the surface dries out, and then it's too late. So notice here that we have, in this case, a plastic chamber that covers our tray. It also has a little place here to open to allow moisture to escape, or you can simply remove that occasionally to remove some of the condensation that develops in there. Once the seedlings have started to emerge we're going to want to remove that cover. So now temperature. Temperature, as I said, is really important depending on the season of the seeds that you're growing. And so the best temperature for many of our warmer season seeds are 65 to 75. For our cooler season, they prefer about 55 degrees. Such things as your flowering larkspurs and snapdragons, your edible peas, cabbages, broccolis, and cauliflowers, which of course that season has just passed. Hopefully you have started and already have grown on your seedlings and transplanted into your garden for your spring, summer edibles. Now, some places where we can then provide this heat would simply be on the top of a refrigerator. We get heat coming up from the back of the fridge, and it tends to make a warm zone. The top of a hot water heater would even be better. It's kind of limited in space. But the better way to go would be to purchase one of these seed heat mats for 20 to 25 bucks. They do sell a thermostat that you can get, but I found that that really isn't necessary. The mat by itself works just fine, and it's very low energy use. It provides in the proper temperature to get these seedlings off and running, maintaining that even moisture that you need all along the way. Okay, now if you're growing in the garden outdoors in the soil, you want to make sure that the soil temperature is proper. You don't want to plant seeds too early in the spring or too, in the wintertime there where it's too cold and they're not going to germinate. We want to wait for the soil to be warm enough. Otherwise, again, they could just rot in place or not germinate at all. So alternatively then for winter, make sure that the soil is cooled down and it's cool enough. So we've grown, our seedlings have emerged. We had 3 to 4 seeds per space. We want to go through and thin out our seedlings. Now the time to do this is when we get the second set of true leaves. And a true leaf is the leaf just like the mother plant. The first leaves that you see emerging on a seedling are the cotyledon leaves, which are not like the mother leaf. And so you want to wait for the second mother leaf. You want to thin these out. How do you thin them? You can pull the seedlings out, but in doing so, you may damage the roots of the sister plant that's there. Alternatively, you can take a small pair of scissors and get in there and actually clip right at the soil line and remove that extra seedling. Always taking the strongest one. So we're growing these on then. We've got that second leaf. We want to start providing some food for those plants in the way of either a mild worm tea or fish emulsion and give them a little nutrients. Very important, as soon as these seedlings do emerge, I like to get my artificial light right in there immediately. And we're going to talk about lighting in just a moment. But if you wait too long on that lighting, then those plants are going to be spindly. And we want to prevent that. We want to have short, stout, strong plants. So as these are growing then, and they have gotten to a size after a few weeks, we might want to consider removing them from the plug tray or from the container that they're in and moving them up one size to one larger container. We never want to overwhelm a plant and go from a 2-inch to a 6-inch, for example. We want to always move one size up from a 3 to a 4 and one size up so that it can fill in that root system and won't be overwhelmed. So we want to use sterile containers, sterile potting mix that's appropriate for those particular plants, whether it be, well-drained media or a more tropical type of a media, choosing that appropriately. We're providing the proper lighting all this time. And we're going to grow them indoors for about 6 to 8 weeks, at which time they're ready to move outdoors, to acclimate to the outdoors before we plant into the garden or sell at a plant sale. Now, while I'm growing these little seedlings indoors, I have my setup where I have a little fan that actually blows on the seedlings and blows across them. This does 2 things. It moves the stems and strengthens the stems, and it helps prevent any airborne diseases. So strongly recommend a little fan blowing on your seedlings as they're growing. They may dry out a little bit quicker and require a little bit more moisture, but you're going to have much greater success. So lighting. And this is something that's really important with your indoor growing. Of course, outdoors, sunlight provides all the light that you need, but indoors, and it must be, as I said, really right in there and up close to these plants, and I'll tell you how close in a moment. First of all, the color spectrum. For indoor growing, the first vegetative state of your plant, the first vegetative growth of a seedling needs the blue spectrum of light. This is 450 nanometers. Now, when you're growing something indoors that you expect to go forward and allow it to flower, for example, people that grow marijuana indoors, they're going to need to have the red light spectrum for that flowering phase. But typically, for most of us that are starting things indoors, we're simply getting them started and moving them outdoors, and the blue light is all that we need. So, the timing that we want to provide for these seedlings is 14 to 16 hours, and setting them up on a timer so that the light can turn on and off automatically. You know, taking note of I like to take note of the price of electricity. You know, they just changed the time frame now from 4 p.m. to 9 p.m. is the heavy period, and they charge you the most right now. So I always start my lighting on my indoor plants at 9 o'clock at night, or maybe I start at 8 and run until 11 the next morning so I can get my full hours in, and it's at a cheaper rate. Now, the brightness or the color of your lights, which is a daylight color, we want it to be between 5,000 and 6,500 Kelvin, and that's the color, and that would be a daylight coloration. And then the brightness of the light that we're providing needs to be 2,000 lumens or more to have the proper brightness for these to grow. Outdoors, of course, I said sunlight, it provides all that you need. And you can find this information on bulbs. If you're shopping on Amazon, for example, you'll find this information listed, and this is specifically what you're looking for. So your options on lighting. We have this compact fluorescent lighting. It could be this screw-in type here, low wattage use. We can have the regular tube, fluorescent tubes, the T5 or the narrow ones that are like ½ inch, the T8s, I think I've got it right here, are the 1-inch size. So the fixtures here is much smaller. It has a reflection in there that it'll project that light down on the plants. But you need to have these lights within 4 inches of your plants. And as the plants are growing, you need to be able to move them up so they're constantly within 4 inches of those seedlings. And those plants may be, during that 6 to 8 weeks, they may end up growing up to be about 8 or 9 inches in height. So you're constantly moving them up. So having some sort of a rack or a way to hold that light fixture and move it up. And there are things available for that as well. Now, down here at the bottom, we have the LED lighting. And I've pretty much entirely gone to the LED lighting. You don't risk breaking the fluorescent tubes. The difference is that with these multi-light fixtures, you have so many bulbs there that you can keep this fixture, or you need to keep it about 21 inches away from your plants. And again, moving it up as need be. The energy usage on this is very, very minute. It's actually equivalent or less than fluorescent tubes as well. So the LED, there's even ones where you can tune in the lighting from the blue to the red, depending on what your needs are. They have a dial to tune that in. So very versatile in the LED lighting. So we've had our plants planted, our seeds planted, and they're emerging from the soil, and all of a sudden we notice that they're getting mushy brown and keeling over right at the soil line. Well, this is a disease called damping off. And it may have been because it was too wet. It may have been that those seeds were infected when you got them. 3 fungal diseases comprise damping off: Pythium, Rhizoctonia, and Fusarium, and combinations of those. Now, it may be that a seed doesn't even emerge from the soil, and before it even emerges, it becomes infected. And you don't even know, and you say, boy, I had poor germination. Well, it may be that you had a damping off problem as well. So this is especially on young seedlings that are less than 4 weeks old that you'll notice this. And this is where removing any covering that you have over those seedlings as soon as they emerge. Getting that fan growing on them will help prevent those fungal diseases from setting hold at all. Now, I mentioned earlier about a seed cover that you can use, and that is a sphagnum moss seed cover, and it's called No Damp Off. Now, you can find it on Amazon, I believe. I think Home Depot has it. You have to order it online and either pick it up at the store or have it mailed to you. But it goes a long ways. You put a light dusting over the surface of your seeded area, and the chemical that's released from the peat moss itself, I'm sorry, sphagnum moss, prevents damping off, and it works really well. Alternatively, here's an old-fashioned one, chamomile tea. You brew up a strong batch of chamomile tea, you let it cool down, and you use that to water these seed trays. Chamomile tea, if you look it up, has lots of factors to it to prevent diseases from happening. So there's an alternative as well. So we've grown these guys now for 6 to 8 weeks. They're ready. You've moved them up perhaps once or twice into a larger size container. They're well rooted. They're nice stocky plants. You've provided proper lighting. You've been fertilizing them with mild fertilizer along the way. So about 2 weeks then before you want to take them outdoors to either acclimate the containers for later sales or otherwise, or planting into the garden, we need to harden them off. And now here in Southern California, the conditions here are so mild that I find I can get away with 1 week of hardening off usually. But the whole purpose is that you're gradually introducing these little seedlings to the outdoor conditions. They've been growing indoors in a protected, high humidity, protected temperature situation, and all of a sudden you're throwing them outdoors to cold nights and hot days and full sun. So what we want to do is gradually expose them to these conditions. The first few days, they'll be brought out into the shade, and then part of the day and brought in at night, and then till gradually they'll be outdoors all day in the shade. And then ultimately, part of the day in the sun, part of the day in the shade, ultimately all day sun, all day and night. And at the end of that 2 week period, you can then have your hardened off plants that will do fine when planted into gardens, and they'll be protected against insects and diseases and so on that might have been an issue otherwise. Okay, so hardening off is important, but we can slide on that a little bit sometimes. So now we talked all about starting things indoors, and I like doing that because I can really control the conditions. But outdoor germination, and there's some things that are better grown right in place. And edibly wise, of course, your root crops, also things like peas and beans prefer to be planted in place. So in doing so, we want to prepare the area, and might be that we need to add some organic material to that soil in preparing it. I personally am going a little different way now, and I'm going with no-till gardening. At my garden site that I have at Mt Sac, we have 15 raised beds that are 4 by 16 each, and we are now evolved into a total no-till gardening for lots of reasons, and that's a whole nother lecture. But we want to prepare that area of organics if we need it, maybe a little fertilizing, watering that soil in so that, again, when you plant the seeds and you do water, your seeds won't fly everywhere. So pre-moistening is helpful. Planting them at the proper depth, 2 to 3 seeds per planting hole, and then using your fine soil or compost or worm castings as a seed cover. You could even use your sphagnum moss out there as well. And then using a fine hose end nozzle. I like to use the Dram Redhead, which is a really fine watering head for your watering wand, and it doesn't disturb your seeds then. You want to protect your seeds from birds eating those seeds, from sow bugs eating the seedlings when they emerge. So one of the things you could do is water them in well, and you could cover them with a piece of cardboard or a piece of carpet, and then check that water regularly. And then once the seedlings have emerged, then uncover them. But that will maintain your moisture level, particularly as hot as it is now outdoors. It's a tough thing to do. Then, of course, you're going to want to thin those seedlings out, just as we did with the indoor seeds, and grow them on appropriately. So now, I'm going to show you here briefly a database here. First of all, here was the handout that I sent you guys, and you should all have this, which just kind of goes through all this and you can have for your notes. But here is, let's see if I got this right. Actually, I'm going to go to a different place. Hang on. Or maybe I won't. Okay. No, but I'm going to show you a seed saving guide. And this seed saving guide should be here right about now. And this is something that I've also, I think, have available to you. I've given you this link, and it's an extensive guide in saving seeds from your garden. First, how to successfully grow things on, when is the appropriate time to save them, how to clean those seeds, how to store them properly. And it goes through many, many different varieties here and how to do this. So it's a great handout for you that I'm sure you'll be able to use. What I'm looking for, though, I'm not finding here, which is another handout, which actually it's a website that you're going to, which is the tomclothier.hort.net. And at this website, you will see a listing of plants, annuals, perennials, trees, and shrubs, mostly ornamental, but it's going to give you great information about how to germinate those seeds best, what temperature, what type of media, if you need to do special treatments for hundreds and hundreds of seeds. The Backyard Gardener also does that same thing. We'll give you that information. So between the two sources, I think it'll be a wealth of information. And then just showed you the seed saving guide that you'll have as well that you can utilize. Okay. I'm not going to try to go to them here because it usually doesn't work for me to leave my Zoom and try to click on it here. So I'm not going to do that. All right. So let's go forward then. Hopefully you're going to have any questions that you chat in the chat area that we'll be able to discuss later. Asexual plant propagation with cuttings, division, grafting, layering, specialized structures, and tissue culture. Of course, for you guys, I'm sure that a lot of air layering and a lot of grafting that you do. I do have a couple of things that we'll take a look at here as time permits on such. So with your cuttings, timing is important, and then I'm going to talk about some basic techniques and then take a look at some specialized forms of the stem, leaf, and root cuttings. So herbaceous cuttings from plants that are fleshy, coleus, for example, can be taken at any time of the year when they're actively growing, which for us is any time. And they're very easy to root. We typically don't even need to use any rooting hormones on those. Softwood cuttings, though, are taken usually right now, April through May, depending on the weather conditions. Of course, with global warming now, our winters are shorter and springs are warmer. And so we're taking these sometimes in late winter versus in spring at all. We want these to be tip cuttings from an actively growing plant that's a healthy, actively growing plant. And we want to take these and we want to be able to bend them and have them snap. So they aren't going to be so soft that they're just bendable. They want actually to snap when we bend them, okay? And that's important on softwood cuttings. Otherwise, you're not going to have very much success with them. They're just too soft. So during this process, we, of course, will need to prevent disease. We want to minimize water loss from these cuttings, and we want to encourage rapid root formation. That's our goals. So, of course, with disease prevention, and this can be used with our seed starting as well, if we're sterilizing our seed trays, if we're reusing any containers for our cuttings, and we want to sterilize them. You know, we always used to use bleach, and they used to say either a 10 or 20% solution of bleach, but we've evolved now into instead using products like Lysol. And Lysol is highly recommended. Now, first you want to, of course, clean your pots, wash them, and removing dirt and soap and washing them down, and then spraying them with your Lysol, allowing them to dry, And in some cases, you may want to rinse them before you actually use them again. There's another product out there that some of you might be familiar with, Physan 20, which is an algicide, fungicide, bactericide, and viricide, which is commonly used in horticulture. I used to use it at the Huntington between crops in the greenhouse to spray down the benches and then wash it all down so that I made sure that things were sterile for my next crop. And of course, we always want to use new soilless media when we're dealing with our root, with our cuttings, tip cuttings. So the idea here is to minimize water loss while we're going through this process. So the plant that we're taking our cuttings from should be well watered the day beforehand so that the cuttings will be well imbibed with water when we select them the next morning. They should be taken early in the morning before the sun really hits that plant. These are going to be the strongest cuttings to use in for rooting. We should process them immediately if we can. And if you can't, you can put them in a plastic bag with wrapped in a damp paper towel, or simply store them in a cup of water for a bit before you're going to actually stick them, as we call it, when we actually put them into our media to root. Some of your, of course, hardwood cuttings would be taken. Of course, for us here, we don't really have much in the way of hardwood cuttings. We don't have cold winters, but yes, there are some plants that we can collect when plants are dormant and use those as hardwood cuttings and can be stored for a period of time before we do use them. We'll talk more about those in a few minutes. So the media that we want to root these cuttings in needs to be sterile. The containers are sterile. Something like 80% perlite, 20% peat, or there's that cocoa peat, for your standard softwood cuttings. But if we're dealing with other plants that maybe don't want to be as wet, maybe we're going to use a more of a coarse sand mixture, or if it's a cactus or a succulent, then we're going to use a fine gravel or coarse sand media. Again, there's different medias that we use for different types of plant material. And that is something that comes with experience. You can find a lot of that information online by Googling your particular plant. And when I Google and want to get questions on a particular plant, I always indicate, what the plant is, where you're at, Southern California, the time of year. Those three things are really important to get the information that you want to get good information on that particular plant material. So we want to moisten this media before we stick our cuttings in there to root, pre-moisten it. And of course, you know that on your tip cuttings, you have your nodes where your leaves emerge. You have that space that's in between the nodes that we call the internode. And in some cases, that internode space is very, very close together. The leaves are very close together. But when we do are propagating these cuttings, we like to have two nodes stuck into our rooting media if possible. Okay, so we're going to take this tip cutting, we're going to make a clean cut underneath this node, remove these leaves, this set of leaves, remove the second set of leaves, and then go forward. So here we are. We have a tip cutting here where we're going to remove these two bottom sets of leaves, as we just said. The tip cuttings should be about 4 to 5 inches in length. And of course, if you're doing something commercial or we have a whole group of plants that are going to be displayed together, they should start out the same size so that the plants, when they're displayed, will also be the same size if possible. It just makes a better appearance in marketing. So you remove the two bottom leaves. The leaves are where moisture is lost while we're trying to initiate roots at the nodes. So if we reduce the number of leaves, we reduce the size of the leaves. So we're typically removing 1/2 to 2/3 of the leaves from this tip cutting. And as you can see over here, we've actually taken this one leaf and actually just cut it right in half to reduce its size and reduce loss of moisture while we're trying to initiate roots. Hormones. Now, the hormones are a synthetic form of auxin. And of course, auxin on a plant is, for example, on a tree. The tippy top point of that tree is where it has rapid cell division, and that's influenced by the hormones that are there. and that hormone is actually auxin. So they discovered this, that at that point of rapid growth on a plant, that they can synthesize this auxin and make it synthetically, and then we can utilize it to initiate roots much faster than we would have otherwise. So these rooting hormones come in different forms. They come in powders that have been around for decades, more recent gels and liquids. If we're using these, they come in different strengths as well. If you're simply working with a softwood cutting or a herbaceous plant, 1,000 parts per million. If we're doing a semi-hardwood cutting, which is later in the season, 3,000 parts per million. If we're dealing with hardwood cuttings, then we should use a hormone that's about 8,000 parts per million. Do we always need to use hormones? No. Particularly with that herbaceous material, we generally don't even need it. With succulents, no, we generally don't need it. So again, it depends on the plant material as to whether we use it at all. When we use our hormones, we never want to stick our cuttings into the container holding the hormones. We want to pour some of the hormones into another container where we'll be achieving it for our cuttings. Now, on these liquids, they're pretty cool because they come in a concentrate, and you add water to form your different concentrations. So you can make a very small amount of this, and depending on what you're dealing with. The gels are great because they adhere very well to the tips of those cuttings. And the powder, sometimes we'll moisten the tip before we touch it into that powder so it will adhere better. So I said we're trying to reduce water loss during this whole process. So we have our tip cuttings that we've trimmed up and removed a half to 2/3 of the leaf size. We've removed the leaves from the bottom 2 leaf nodes. We've maybe used rooting hormones if necessary. And we've placed this tip cutting into the rooting media. Now, we want to actually make a hole with a pencil or a chopstick to stick that cutting in. If you just try to force it in, you're going to break it, or you're going to knock the rooting hormones off. So make a hole in the media and then force that media around it. And as you can see, we can place more than 1 cutting in a container. Sometimes, depending on the container, 3 or 4 or more. And then we want to provide an environment for this cutting to remain moist so it doesn't dry out. So in this case, we've simply covered it with a plastic bag. Remember, we pre-moistened that media, and we want to use something to keep the bag from touching the leaves of the cuttings. So we can use chopsticks or pencils or the like. Alternatively, this plant that's placed in this media could be set on in a chamber that maintains a high humidity, or on a misting bench that actually sends out a fine mist on a pre-programmed scheduling time schedule. And of course, during this process, lighting isn't necessary. So subdued lighting for these guys. We're initiating rooting, and for rooting, we don't need to have a lot of light. So now after 2 to 6 weeks, we can remove our bag or remove it from the chamber and check to see whether we have roots. And we can simply tug a little bit on that cutting. And if there's resistance, then chances are you're good to go. If you're not quite sure, you could dump the contents out and check it and see. And if it's not well rooted yet, you could pour everything back together again and place it back in. You don't need to hormone it at that time. Now, if after 8, 9 weeks, if you have no rooting taking place, chances are that you failed. And it may be because the timing was wrong, the process that you did was wrong, the hormones weren't right, moisture, a lot of different factors. If your cuttings have died back, then you may have had a fungal disease, or they were too wet. So keeping a journal is really important, especially with any seed starting or cuttings, so that you can make note of what you did do. So then if you have successes, you'll know your successes. If you have failures, you'll know what to do different next time. Okay. So we now have cuttings that have rooted successfully, and we want to gradually acclimate them. So if they're in a plastic bag or on a chamber or on a mist bench, we want to gradually cut open that bag or remove them from that mist bench. They'll still be in the greenhouse. The humidity will still be high, and they will then gradually acclimate. after which time we're going to, after about a week or so of acclimation, we're going to take those individually rooted cuttings and pot them up into an appropriate size container, a sterile container, sterile potting media. And remember, you don't want that container to be too large. And then gradually, as it's growing, you'll move it up to larger size containers. So your semi-hardwood cuttings are taken from the current season's growth after the wood has matured. So we talked about those semi-hardwoods where it was early in the season and just when that little tip cutting was snappable. Well, now we've gotten to a point where the wood, the bark, let's say on this little cutting, has firmed up and the leaves are full size. So this is typically midsummer, which again, the timing on that is changing as the temperatures and climates changing, into early fall for most plants. Many different evergreens, boxwoods, hollies, rhododendrons can be propagated this way, okay? And at this time of year, using the same methodology as we did with our semi-hardwood, maybe a stronger rooting hormone, that'd be the only difference. Hardwood cuttings, typically the most difficult to root. So these are shoots that grew the previous summer and they're cut during the wintertime or early spring when the plant is still dormant. And of course, for us here, sometimes we don't even have dormancy, right? You know, our fruit trees here, that's why we try to at least buy low chill fruit trees and grow those, or our subtropicals are probably going to be a lot easier to grow tropicals and subtropicals in the future as global warming moves north. So the wood is firm, it doesn't bend easy. There's deciduous shrubs and fruit trees and evergreen, needled evergreens that can be rooted from this type of wood. So the bark has hardened. There's certain ways that we're going to treat this. So let me show you what we're going to do here. We have to be careful with needled evergreens that that we are careful about water loss on those, okay? Because they can very easily not have enough water and lose all of their needles and fail. So on a hardwood, a typical hardwood or a fruit tree that's in a dormant state that we've collected and we're ready to root, we've maybe stored this for a bit in a cool refrigerator for some time. And it's time now to root these on. So we want to trim off the bark to expose what's called the cambium layer right underneath here, which is the actively growing part of that plant. So we've trimmed that off and we will then proceed with our rooting hormones and our media and root these on. Now, on an evergreen, we did a little bit differently. Instead of cutting off or shaving off some of the bark, we simply cut into the bark down into the cambium layer and proceed with our rooting hormones and rooting. So these hardwood cuttings then have been treated with the hormones. We've made a hole, we placed them in there, forced our media around, and now in many cases they will have no leaves, they're leafless. What oftentimes happens is they will start to leaf out before they even root. So you need to then be checking them regularly for that time when they've actually rooted. One of the other things that oftentimes occurs on hardwood cuttings is that we get what is called callus, where we've made that wound. It's kind of like a scab that occurs on that wounded area. And sometimes we don't get rooting unless we get callus that forms. So the two go hand in hand. So let's proceed here with some other types of cuttings that we can do. Here's an ivy. Of course, I don't know why we'd want to propagate ivy, but we can take this ivy and lay it across our media with the nodes slightly buried into that. In this case, it could just be potting soil. And you'll notice then that we're going to get roots that occur at each of those nodes where the leaves are along that stem. So at that point in time, we can actually cut them into individual pieces and plant that rooted little leaflet in its own pot and start a whole new plant. Here's a case of, could also be done with ivies, but with geraniums, philodendrons, peperomias. We can take a stem from this plant and we can actually cut them into individual pieces, take those individual pieces, treat them with our rooting hormone, place them into our media, And then ultimately, we're going to have a little baby plantlet that forms with a new root system, at which point we'll want to remove that old mother leaf and allow that new little leaflet and plant to grow on, and replanting it into the appropriate size container as well. Leaf cuttings, and we can take the single leaf from certain plants, succulents, begonias, African violets, peperomias, and it can be done a couple different ways, but we can cut right through the vein on some of our heavily veined leaves on the back side, lay that leaf across the surface of our media, and actually pin the leaf down so that leaf with a cut will have constant contact with the soil, and we're going to get roots that emerge at that point. We can do it another way. We can actually cut that leaf into individual pieces and stick them straight down into that media, and we're going to get roots that will form at that point. We can take a Sansevieria plant here and cut a leaf into a section or a segment and stick that into our rooting media, and it will root and form a brand new root ball in a new system. But the thing is with this is that the baby plant, in the case of the Sansevieria, will not be identical to the mother plant. And some of these others as well, they might have some differences and revert back in their genealogy to a different plant than you originally saw in the mother plant. Okay. On an African violet, here's an example where we can take a leaf from an African violet, cut the petiole, which is the stem, to 1 inch in length, dust it with a rooting hormone, make a hole, place it, and we are going to get roots that form, and we're going to get maybe 5 or 6 little baby plantlets that we can separate, each of them having its own root system, pot them up, getting rid of the old mother leaf. But note that in this case as well, these baby plantlets will not be identical to the mother. The flower will not be the same. It will revert back in its genes to some previous version. Okay, but you might get something great. So root cuttings. This would be done, for example, on a philodendron where we have a very fleshy type of a root. You'll oftentimes see a philodendron roots that go actually outside the pot and grow outside of the pot. You can take that root and cut it into a section and lay it across the surface of your media. And you'll notice then, and we don't need rooting hormone, or if it's a large size of these roots, you can stick it down into the media. In both cases, boom, you're going to get your fine roots that form in a new leaflet. Notice that they did cover this up to maintain the humidity in there as well. So then we'll take that individual plantlet and pot it up, and you'll be on your way. So division. I mentioned Sansevierias. If we take a leaf section, the result will not be identical to the parent. But in division, it will be. So we can take a growing Sansevieria plant and divide it and take a section of the existing plant root and replant it, and it will be like the parents. Same thing with your African violets. We can divide that mother plant, and the baby plants will be just like the mother plant was. There's other things like grasses, hostas, irises, ferns that are divided, daisies as well. And when we do divide our plants in the landscape, we typically want to cut those plants back to just within a few inches. And timing is important. Oftentimes this is done at the end of the growing season in the fall when the plants are going to go dormant. We cut the plants back and to just a few inches, actually dig down and remove the entire root section and leaf portion of that plant. We need to fill in that hole that we've left there so that the remaining plant can continue to grow. And the portion that we have divided from the mother plant will repot or plant out back into the landscape in the appropriate manner. So as I said, some tips for this, cutting back your foliage will make it easier to handle. Also, and will then put more energy into the rooting of that plant once you've replanted it versus trying to support leaves on that as well. So in removing most of the leaves, it will then initiate more rooting, which is what we want to do. We might want to, if it is warm or hot when you're doing this, you might want to shade this divided plant for a few days and watered in well to acclimate it. So now, of course, what I'm sure many of you are familiar with is grafting. And this is, of course, has been around for 1500 BC, and it's joining together two or more plants so that they become one. And this is commonly done with ornamental plants, with fruiting plants. Many different things can be treated in this way. So we're going to do it to combine two plants that are related to each other, but are different, so that we can have two different things on the same plant. We could be grafting to create an unusual new growth form. We could be trying to make a novelty plant. We might be trying to change our fruit varieties, maintaining our rootstock, but adding a new scion to it to change the variety altogether. Or we might be wanting to repair a wound in a limb, in a branch, or in a root itself. We can actually do grafting on those as well, just to repair a wound. So some of the terms that I'm sure you're familiar with, of course, is we have our rootstock. And rootstock is usually a pretty tough material. It's something that can handle either a heavy clay soil or diseases in the soil. So we're using it for a purpose, for that very purpose. It's going to be tough and allow us to attach something to it that then might not have done so well in that particular soil. So the piece that we're attaching to the rootstock is called the scion. That scion should have 6 to 8 buds on it if possible, growth buds. Sometimes we're going to add another piece in between that's called an inner stock. And in that case would be something like a tree rose, for example, is done by 3 pieces that are grafted together. So some of the tools, of course, you're familiar with the different types of grafting knives or devices that we'll see in a moment. I love this wax grafting tape. In the old days, we used to use the graft rubbing bands, and in the video today, he does use those sometimes. I was taught to use black electrical tape. The thing about that is, though, you do need to remember to remove that after a period of time, because it doesn't stretch the same as this wax tape does. And the use of rooting hormones is optional with grafting. In some cases, it does improve your take, but oftentimes it just isn't necessary. Again, with a branch of a tree or a trunk of a tree, we have the outer bark. We have just beneath that, we have the area, the growing area, the cambium layer, which is the portion that we have to have attached to the new portion that we're combining it to. We have the xylem and the phloem that bring nutrients up and down from the plant itself. But it's very important that we have this close, intimate contact of the cambium layers when we are grafting. So some commonly grafted fruit trees. I know that many of your tropical and subtropicals as well, but apples, pears, peaches, plums, cherries, citrus, grapes, avocados, olives, mangoes, apricots, many different nut trees. Of course, nut trees, we typically don't have cold enough weather here to grow nuts in our region. Ornamentally, could be magnolias, roses. Peach tree down here has multiple peaches on one tree. This is a bougainvillea in the middle down here that has different colored bougainvilleas. The last couple of years, it's become popular to graft tomatoes, utilizing a tough rootstock and an heirloom variety that typically doesn't do well for diseases. So we can graft an heirloom onto a tough rootstock and have healthy heirloom tomatoes. Here's a novelty here, utilizing potato as the rootstock, to which we have grafted tomatoes. They're in the same family. They're both in the Solanaceae family. So once your tomatoes are through and you've harvested those, at the end of the season, you can dig up your potatoes and enjoy those as well. So there's different types of grafts that are utilized. Some of the common grafts are the whip and tongue, the cleft graft, and this is oftentimes if we're changing out a variety of a plant in the ground and we want to change it to a whole new variety, we're going to use the cleft graft. Maybe starting with 2, it may end up with just 1 variety, but hopefully we can have 2 or more varieties that we're grafting onto that rootstock. There are even some special devices that we can buy that make cuts for us, special grafting cuts that we can then join together that maybe give better strength than you would get otherwise. Here's a side cleft graft. So different plant materials do better with different types of grafts depending on the type of material and the timing that you're doing it. Here we see some of these, of course, budding is actually a form of grafting. Some people might want to tell you different, that it's its whole thing, but it's actually a form of grafting. And there's actually special tools, budding tools, but we're actually removing the this growth bud from one plant and inserting it on another. Here's a case is a citrus where we have prepared the area. We've taken the bud from one and we've inserted it into the other here. Here's a rose bud that has been grafted here. This is a method that's a simple, I find a very simple method of propagating, easier than grafting myself. There's different ways that it's done. Marcotting is the terminology. So we make an injury on a branch of a actively growing tree. And at the point where we make this injury, then all the energy from the leaves, the food, the photosynthesis that takes place, the food that's formed, it stops at this point. So what we can do then is we can initiate rooting where we've made this injury, because the food that is stored up then will initiate the rooting. Now, again, it's done in a lot of different ways. Here, again, is ivy where they've made an injury and they'll wrap it. And here is on an actively growing tree where we've made that injury. We take sphagnum moss, the stringy moss, soak it in our liquid rooting hormone, and then we have to open that wound up and shove some of that sphagnum moss in that cut, wrap it around the branch, cover it with plastic, cover it with foil, and in about 6 weeks, roots will form. Now, it's done in different ways. Nowadays, I see a lot of different methods. Okay, yeah, we'll just do it from here and won't go to full screen. Okay, sorry about that, guys. So we can do other layerings. An actively growing plant, a rose, we can bend over the canes and touch the ground and get rooting to occur on many different plants in the garden while they're actively growing. And then once it has, we can cut it from the mother plant and have a new plant to dig up and replant. Tip layering, raspberries, blackberries, just the very tip touching the ground is going to root. And again, those blackberries are going to continue. I know I have 30 feet of blackberries in my backyard that want to keep going, and I have to stop them from advancing. Here's an actively growing plant that we just have the nodes in a container with potting soil, and in each of those nodes we will get rooting. We then can cut and separate them from each other and have a whole new plant growing as well. Specialized structures, bulbs, and the like here. Let's take a look at those. True bulbs, daffodils, tulips, they will actually have a basal plate at the bottom, and then from that point, the new little bulblets start to form. The original bulb dies back, and the new bulblets take over. With a corm, we also have that basal plate, and we get the auxiliary buds will start to form at that point. The original corm then dies out, and the new auxiliary buds become new corms. So crocus and gladiolas. Tubers, like potatoes, have no basal plate or one originating growth point. Roots grow from all over. Tuberous roots, like sweet potatoes, dahlias. If we're dividing those, we must make sure that we have both a root and a stem, otherwise we won't have success. Rhizomes, like irises, they have a root system that advances and continues to grow, and the old portion tends to die off. So they have to be divided occasionally and cleaned up, removing the old portion from the new, replanting and allowing to continue to grow. Tissue culture, briefly, is where we can grow thousands of new plants from a small portion of a plant. They take a piece of plant and use particular hormones and chemicals. And these are done by companies that have developed these trade secrets, and they don't share this information readily. And they then are adding these different hormones. First, they're going to get a single cell from a leaf, a root, or a flower, and the growth regulator will make it to start to grow. Then they have to change to a different growth hormone that will initiate rooting, and then we can have from the single cell a plant, a rooted plant that will grow and get thousands of plants from a very small plant portion. This is done, of course, with, now we're going to give you the link for tissue culture if you want to watch that video. It's pretty interesting. Some of the plants that are propagated by tissue culture, African violets, chrysanthemums, orchids, dahlias, bananas, eggplants, tomatoes, hops for beer, are readily propagated by this method. And more house plants all the time. You know, it's done in a way where diseases are prevented and they can get an exact duplicate of the parent plant. So we made it, guys. A few issues and problems there, but I'm open for, let me change the view here to questions. If anybody has any questions, or was anything in the chat area that needed resolve.
Thank you so much, Stephen. That was super thorough and
A lot of stuff. Hard to get it in a short period of time. It's usually a 2.5 hour lecture or so. So
yeah, this is definitely a solid propagation 101. Definitely can take any approach. So we had a question. Just so folks are aware, there is the handout in the chat window. You can pull up the chat window to find that.
so there are all the links that you need for those different sources are there as well for you.
Yeah, click on it and it'll download a PDF for you. One question, besides the LA County Master Gardener planting lists that are broken down by month, do you recommend any other resources pertaining specifically to LA County?
You know, that list that Yvonne Savio did is so old now, and the weather climate has changed, and therein lies the problem. I use that list still. I've been using that in my classes, but I find more and more that our growing seasons are changing and the planting times are changing a little bit on that list. But it's still a very, very good list that really does give you, oh, we got a different view there. It gives you really good information.
Right on. John, you had a question. Do you want to read it, or would you like me to? You're muted, John.
No, go ahead, Ty.
So on mycorrhiza, do you promote that as far as an inoculant, and at what specific applications?
Well, yes, for growing plants, it's not for the seed state or for cutting state. It's for later growth, of course. And this past year, I've been doing a lot of research on mycorrhizae and with trees and plants and how they communicate with this system of that provides nutrients and all kinds of great things. So there's some wonderful videos on that as well out there.
Right on.
It's for later growing, not for the initial stage.
Okay, newer questions. Could you please explain how the leaf cutting doesn't produce identical plants?
You know, it's just one of the anomalies in this world. Just like, you know, we have our hybrid plants and we have heirlooms. The heirlooms give us, you know, the hybrid seeds do not come true to the parentage. It's you know, has a lot to do with the growth and the genetics of it.
But wouldn't leaf cutting be asexual propagation?
I think that it would be, but on those particular, that way of doing it, they don't come true.
Okay. Right.
I'll have to look into that further and find out why that is exactly. I couldn't tell you.
Okay. Another question, if I was planting black goji berry from seed, what temperature would be best? It's cold tolerant and grows in cool weather. And would the humid incubator technique work for that?
Yeah, again, seasonal timing is the right thing to choose the right time and temperatures for particular seeds. So it probably wouldn't need any heat to initiate growth on that. You have to be careful though with some things growing in a chamber that it's too wet or too humid, and getting the right balance on that. I'm not familiar with that particular plant, but I would Google that and find more information about what people have done and what successes and failures that they've had, and then track what you do. And you may have to try a couple different techniques, and you can even do different techniques at the same time and see which one works best for you. That's what I usually do. With something
Buckshot approach.
Yep.
Charlie, you had a question. Would you like to ask it?
Yes, thank you. John McNeil and I have been working on air layering my lychee tree, and we follow the steps of getting the roots to form. But when we try to cut that branch off and put them in the pot, those roots are always very, very fragile, and they will fall off with just the touch of a feather.
Wow.
And it also seems as though those roots never form anything that's a substantial root, like a tap root. So what do we do?
So now, what method are you using for that air layering? Are you wrapping up with sphagnum moss, or how are you going about it?
We're wrapping them in a plastic bag with, I believe, peat moss. If you've seen the DVD that our club has produced, we're doing it pretty much the way Roger Meyer showed.
I haven't. Why don't you try it with sphagnum moss? The sphagnum moss will hold together. The roots can grow through it, and it'll give them support so that when you do cut it off from the mother plant, it'll have something to bond it together better, I think.
Well, the peat moss, if we let it dry out, much like the video showed, and then put it in a pot, we seem to have better success not breaking the roots. But I don't think those roots ever form a long tap root or a structural root. They're just kind of hair type roots.
Again, some plants may be more successful at this than others. And I have not personally done that with that plant. So I'll look into that more myself as well. Okay. But I would try the sphagnum moss because it's a stringy moss. The roots can grow through it, and then you won't disturb them when you do try to plant that up. And maybe you'll have a stronger root system going through that. I don't know.
Okay, well, thank you. We'll give sphagnum a try.
We'll try.
All right, some more questions. Is there a book that itemizes the best way to propagate the many different plants and types of plants?
Yes. Darn it, I'm trying to think here of the book that I used in college, and it was a complete book on plant propagation. There's about 3 different authors, and it's about its 11th version of it. And right now I can't remember. It's been 30 years since I was in school. I can get that back to you, Ty, though. And I'm sure there's others, but there is a good textbook that's out there that has been used in schools that's very thorough.
I had one in horticulture. I believe it was called Plant Propagation.
That
might be it. It had multiple editions.
A green cover. Yeah.
Yeah, they might have changed the cover over time too.
Yeah. I'll get that as well.
Okay. Is there a best method for propagating passion fruit you'd recommend?
I don't have experience with it, and so I would Google and find what others have done, because I personally have not propagated that.
Given that there's semi-softwood or softwood, depending on the time you catch it.
Now, you guys might know the guy that works at Long Beach City College.
Jorge Ochoa?
Yeah, he would know.
Jorge would know.
Ask.
Yeah, he would definitely be, He definitely.
Maybe are different than others, some are done in different ways. So I would ask him about a particular variety that you have.
I was in his classes, and so he would do cuttings and root hormone, drop it into media in a flat usually, and then we would just be teasing them out based on what had roots. Also can start them from seed. He would do crossing of different flowers, and so the seed would be unique, and then once you got the fruit, propagate that.
Sure.
And then I think
A lot of trial and error.
most
And,
of it, yeah.
yeah.
Okay.
Okay, thanks for
Then there was one question if it was the
That's
book.
Hartman and Kester's Plant Propagation. That is the book.
That's the same one we used too.
That's the same one.
It's a classic.
Yep.
Okay. Any last questions? Oh, just wondering, can you graft an air layer anytime other than spring?
I've actually on loquats, I've air layered in the hotter time of the summer and had success. You know, it depends on when the plant is, you know, actively growing is when you're going to have better success. So, of course, heat, protecting it from heat, wrapping on that air layering on a loquat, for example, I will wrap it with a plastic bag and then foil. And the foil then keeps the sun off of it, reflects the sunlight, and keeps it moisture as well.
Martha mentioned that she's had great success with rooting passion fruit cuttings in a 1 gallon pot without even using rooting hormone. So there you go.
And that's the good thing about plant propagation is when, you know, people share information, and there's actually an organization, Plant Propagators Organization, that's just what they do, and they publish information that they have developed and they share.
Right on. Well, I think that wraps it up. Oh, last question popping in. How long can you keep my computer just threw something in front of me. You'll have to read that.
You mean like your how long can you have your refrigerate your scion? You know, usually you want to have collected it in the dormant time and then the following spring use it. You might be able to hold off for another year, I don't know, but best to use it that first spring.
Well, thank you so much, Steve. You Really gave us a good detailed analysis. I love it. I love it. We've got a good reference point now for our plant propagators in the chapter that feel inspired to grow and propagate for our annual plant sale. And
I always look forward to it and seeing you guys, and hopefully we'll be at the green scene next year and see you back out there again. If not yourself, so
that would be ideal. Well, thanks so much, Steve. Yeah, thanks for taking the time out of your day.
Thank you very much.